A'udzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Nahmaduhu wa nusalli ala rasulihil kareem. Class, this is our first lecture, or maybe we can say this is our first introduction. Inshallah, we will going to have the classes in this semester, maybe completely on online, or a hybrid will be adopted. The course is Pharmaceutical Chemistry 4B, Medicinal Chemistry Form 714. Dr. Saira will be the associate teacher. She is going to associate me, or maybe taking few lectures. First, we are going to discuss the course contents. Just I am reading that one. We are not discussing at this level. General properties, chemistry, biological action, SARs, and therapeutic applications of the following, such as sulfonamides. There are a number of examples which we are going to discuss. Prontosil, sulfonamides, sulfopyridine, sulfadimidine, sulfamethoxazole, sulfadiazine, and sulfafurazole. Next one is the antimalarials: 4 aminoquinolines, 8 aminoquinolines, 9 aminoacridines, bioquinoides, pyrimidine analogues, mefloquine, and cinchonalkaloids. Next, we have got the diuretics: mercaptomarine, maluride, thiazide, spironolactone, theophylline, furosemide, acetozolamide, ethacryonic acid, and thymeterine. Antitubercular drugs includes ethambutol, isonicotinic acid derivatives, hydrazide, rifampicin, thioguanine, pyrazinamide, cycloserine, ethonamide, cytarabine, 5-fluorouracil, and dicarbazine. In antiviral drugs, we are going to study acyclovir, tromantadine, hydrochloride, ribavirin, and some other drugs. In immunosuppressants, azathioprine and cyclosporine. In antibiotics, penicillins, cephalosporins, streptomycin, chloramphenicol, tetracyclines, kanamycin, and erythromycin. In this syllabus, few topics needs more detail, such as the sulfonamides, the antibiotics, the diuretics, while the other one maybe we are going to study briefly. The recommended books are Wilson and Gizold's Textbook of Organic, Medicinal and Pharmaceutical Chemistry, Hoy's Principles of Medicinal Chemistry, An Introduction to Medicinal Chemistry book by Graham L. Patrick. It is advisable to use the latest editions of the books, but maybe in few editions some topics may be missing, so you have to get it, and if you are not able to find it, please contact. I will provide you the photocopy or the link for that one. Okay, now we are going to start our first topic, that is sulfonamides. Why we have selected this sulfonamide? As in this case, amino group is substituted, it will become aminobenzene or aniline, but we are not considering the position. It can be substituted at any positions. All of the positions will be the same. But when we are going to substitute an other group, say at adjacent position, then the group or position will be considered as ortho position. And if the group is substituted at alternate position, it will be considered as meta, and if it is substituted at para positions or opposite to the first group, then it will be termed as para substituted or para position or para substituent. It means the ortho, meta and para positions are the relative positions. They are relative positions. They are related to the first group. If the first group is substituted, then we can say the other groups will be ortho, meta or para. Activity of the molecule. Constituent is substituted at adjacent position to the first group, then they will be termed as ortho substituents, or the position will be ortho. If we are going to substitute any group alternate to the first position, then they will be termed as meta group or meta position. And if the substituent will be substituted at opposite to the first group, then it will be termed as para position. Para position. Mind it, the ortho, meta and para positions are the relative positions. They are relative or related to the presence of first group. The adjacent, the alternate, the opposite positions in relationship with the first group. And you know, when these compounds are used in medicine or in medicinal chemistry, the importance of these positions is very clear. Just by changing the position, maybe we are not changing the functional group, maybe we are not changing anything, but just by changing the positions, the biological activity of the molecule can be changed. One of the simple example is the acetyl salicylic acid. In the acetyl salicylic acid, the substitution is at ortho position, that is, we are using the ortho hydroxybenzoic acid. But instead of that, if we are using the meta hydroxybenzoic acid, or the substitution will be at meta position, the activity, the biological activity, the biological function of the two derivatives will be different. Maybe some characteristics, some effect will be the same, maybe the other effect may be retarded, less, or may become more. हाँ चलो
और इसमें से कट
की. You can see here we have used that aniline or aminobenzene. We have substituted another functional group, that is SO2 NH2 that may be termed as sulfonamide group. We have just seen the example of aminobenzene, that is aniline. If another group such as SO2NH2 is substituted on this ring, that is sulfonamide group, the result will be another compound, that is sulfonylamide. In sulfonylamide we have got an amino group which is substituted on the aromatic ring. Another group is the sulfonamide group, SO2NH2 group, which is substituted at the para position of this aniline. Here we can say that if the SO2NH2 is present before NH2, then the NH2 will be at its para position. But in our synthesis, we are using the aniline as a precursor, and then we are synthesizing it by the method of chlorosulfonation. It means, as a precursor, we have got the aminobenzene, and the aminobenzene is chlorosulfonated, and then we have got the sulfonylamide. In the next few lectures, we are going to see the reaction of the aniline. The molecule sulfonylamide is the basic molecule, but any compound which have got the SO2NH2 group will be considered as a sulfonamide compound. Here we have got the sulfonylamide. How we have got this sulfonylamide? Paul Ehrlich, who is considered as the father of chemotherapy, father of drug designing, father of medicinal chemistry, he has discovered the salvarsan, an antiseptic drug, in 1908, and from here we have got the lead. This initiated the use of dyes as antimicrobial agents. What is dye? The dye, the coloring substance. These coloring substances were used to color different substances, maybe the clothes. And you know, one of the pioneer manufacturer is BASF, and still that company exists, and it has got a department for those dyes, or dyes is their primary concern, but they have got the medicinal departments or medicines. Gerhard Domek in 1932, using Prontosil, a red dye, and during his experiments, he has found that the compound is active against streptococcal organisms. But then it was found that prontosil was active in vivo but not in vitro. What does it mean? When they are using the prontosil in vitro or outside the body or outside the living system, the molecule is inactive, but otherwise it is active. We can say that the prontosil is a prodrug, although this problem was resolved in 1935, but before that, in 1933, the compound was used or reported as a compound by which a number of infections were treated or cured. As I have told you that in 1935, we have resolved the problem. We have got an approach that it is a product, it is going to be activated after metabolism in the living system or in the bacteria. Then we have got another evidence in 1937 when we have got the sulfonamide isolated from the blood and urine of patients who were treated with prontosil. From this, it is confirmed that the active compound is sulfonamide. The active compound is sulfonylamide. In this slide, we can see that prontosil, the red dye, is inactive or which is inactive. It becomes active after metabolism or in vivo. The active compound sulfonylamide is the basic molecule, but any compound which have got the SO2 NH2 group will be considered as a sulfonamide compound. Here we have got the sulfonamide. How we have got this sulfonamide? Paul Ehrlich, who is considered as the father of chemotherapy, father of drug designing, father of medicinal chemistry, he has discovered the salvarsan, an antiseptic drug, in 1908, and from here we have got the lead. This initiated the use of dyes as antimicrobial agents. What is dye? The dye, the coloring substance. These coloring substances were used to color different substances, maybe the clothes. And you know, one of the pioneer manufacturing or manufacturer is BASF, and still that company exists, and it has got a department for those dyes, or dyes is their primary concern, but now they have got the medicinal departments or medicines. Gerhard Domek in 1932, using Prontosil, a red dye, and during his experiments, he has found that the compound is active against streptococcal organisms. Streptococcal organisms. But then it was found that Prontosil was active in vivo but not in vitro. What does it mean? When they are using the Prontosil in vitro or outside the body or outside the living system, the molecule is inactive, but otherwise it is active. We can say that the Prontosil is a prodrug, although this problem was resolved in 1935, but before that, in 1933, the compound was used or reported as a compound by which a number of infections were treated or cured. As I have told you that in 1935, we have resolved the problem. We have got an approach that it is a product, it is going to be activated after metabolism in the living system or in the bacteria. Then we have got another evidence in 1937 when we have got the sulfonamide isolated from the blood and urine of patients who were treated with Prontosil. From this, it is confirmed that the active compound is sulfonamide. The active compound is sulfonylamide. In this slide, we can see that Prontosil, the red dye, is inactive or which is inactive. It becomes active after metabolism or in vivo. The active compound is sulfonylamide. The active compound is sulfonylamide. Now we can say that sulfonylamide is the parent compound in our series. The generic name is sulfonamide, that is para-aminobenzene sulfonamide. And as I have told you that just by changing the position, just by changing the functional groups, we have got a number of derivatives. You know, at least more than 5,000 derivatives are synthesized from this molecule. And we have got different derivatives having the different activities. Most of the diuretics have got this sulfonamide group, or the precursor is sulfonamide. The parent compound is sulfonylamide. One of the example is furosemide. Sulfonamide itself is a weak diuretic but have got the diuretic activity. Then by different modifications we have got the more active and selective diuretic compounds. The thiazides, the hydrothiazides, the furosemide are the examples. Thiazide and hydrothiazides are the classes, while the furosemide is the name of one specific compound. A number of antidiabetic compounds. Antidiabetic, the compounds which are used to treat or control the blood sugar level. We have got one of the important compound, that is talbutamide, which is synthesized by modification when this amino group is replaced by CH3 and some substituents are substituted at nitrogen of the SO2 NH2. We have got talbutamide and other antidiabetic compounds. In our lectures we are using the compounds which have got the antibacterial activity. It means here we are not considering the diuretic, not the antidiabetic, but the compounds which have got the antibacterial activity. That will be discussed in detail. We have got a number of compounds which are used as analgesic. Analgesic? The compounds which are going to relieve the pain. The compounds which are going to release the pain. But again, at this point we can say that there are some compounds or a number of compounds which are termed as anesthetic compounds. They are also going to release the pain. Then why we are not saying them anesthetic compound, or what is the difference between the analgesic or anesthetic compounds? Mind it, first we are saying that the compounds are going to release the pain, that will be termed as analgesic. We can say that these compounds may remove the cause of the pain, or they are not going to remove the cause of the pain, but just they are going to relieve the pain. For example, in the cases of fracture, in the cases of bone dislocation, we are using the analgesic compounds. They are not going to provide any relief in fracture, or they are not going to treat the dislocation, but just they are used to relieve the pain. The cause will be there. That's why we have to repeat it after few hours. Say in the case of fever, when we have got headache or body ache, we are using the analgesic compounds. They are not going to relieve the fever, or they are not going to affect the infection or the cause of the fever, but just they are going to relieve the headache or body ache. The cause will be there. When we are differentiating it with the anesthetic, again, in the case of anesthetic, we have got general anesthetic compounds and the local anesthetic compounds. Here we have got the differences in the mode of action or site of action. But in both of these cases, they are going to relieve the pain. Yes, we can say that these compounds are also analgesic compounds. As I have told you before that in the case of fever, we are using, say, any antibiotic to relieve the fever. They are going to affect the microorganisms, but they are not going to affect any other thing. But when they are relieving the pain, they can be termed as analgesic. They are not true analgesic, but analgesic. In the same way, here we can say that these compounds are anesthetic. They have got the analgesic activity. They have got the analgesic activity. We can differentiate between these two, general anesthetics and local anesthetics. In the general anesthetics, the central nervous system is involved. That is going to be depressed at different levels. While in the case of local anesthetics, the central nervous system is not going to be affected. Just a local site, a specific part of the body is going to be anesthetized, or the nerve which is going to supply that site is affected. We are not going in detail of these anesthetics or analgesic, but we should know about the differences of these compounds. The analgesic compounds, the anesthetic compounds, the general anesthetics, the local anesthetics. Very briefly, I can say that analgesic compounds, when we are talking about their mode of action, the analgesic compounds are the compounds which are going to inhibit we can say that the analgesic compounds are those compounds which are going to inhibit the perception of the pain. What does it mean? The pain impulse is generated. It is conducted by the nerve to the central nervous system, but this impulse, this message is not translated, or the perception of the pain is inhibited, and hence we have got the analgesic effect. In the case of local anesthetics, the nerve impulse or impulse of the nerve or pain impulse is not conducted to the central nervous system. What does it mean? The message is not reached to the central nervous system. It is inhibited. The conduction of the pain impulse is inhibited. In the case of general anesthetics, the message or nerve impulse or pain impulse which is generated is reached to the central nervous system, but as the central nervous system is depressed, hence we have got that analgesic effect.